antenna. Hello, welcome to Sir Majesty Easy World Science Channel. Nice to know that you're there. Today I will take a look at this NECO exam, the past exams. Let's tackle them, know the possible thing that we're supposed to answer. We'll pick it and give the likely correct answers there. So it will be nice if I bring alongside the specimen, because okay, I think already highlighted here. So you can see specimen A, it is quite well explained there. This is the list of the specimen, and here is the question paper as it was given to you. So we're gonna do justice to them. I think I have to minimize this. Okay. Should come here a bit. And then this will come here. I will be writing direct. Let me check my pen. Okay, it's connected, of course. So the first question says identify specimen A, B, C, and D. Here you are not required to identify with reason or reasons. You just tell them, as you can see there, that specimen A, you write atlas. Write atlas vertebra. I think I need to reset my pen. Okay. It's not bad. Okay, let me use that side. Okay, just an atlas. If you don't put vertebra, no problem. Vertebra. Then specimen B is just nothing but okay, maybe I think I should divide this screen very well. I think it's better. Yeah, so then specimen B is there, thoracic vertebra. Just write it, thoracic vertebra. Then specimen C is a scapula. Specimen D is lumbar vertebra. Vertebra is singular. When you put it E, becomes plural. That's vertebrae for more than one. Okay, then say the next question says state the location of each of the I think let me use red here to contrast well. State the location of each of A, B, C, and D in the organism that possesses it. It's very simple. The location of atlas is in the neck. Just write neck region, or in the cervical vertebra. Then B is located in the back of the chest, or the chest region, back of the chest, or you say chest region. Then C, scapula, is located in the shoulder, the upper limbs, or you say in the shoulder girdle. Shoulder girdle. Here is shoulder girdle. Or in the upper limbs. Then D, which is a specimen, you call it, is where is it located? In the lumbar vertebra is located in the abdomen. Yeah. So the other one says give two differences between specimen A and D. Specimen A is atlas, D is lumbar vertebra, and I guess you watched my video here before these exams. So this video will help you compare and contrast. Try and watch the video here discussing these bones. Okay, the differences between specimen A and D is just telling you specimen atlas and the lumbar vertebra. There are many differences. I'll give you as many as possible. You can write here presence of vertebratorial canal, that is for atlas. Then here in D for lumbar, absence of vertebratorial canal. Then secondly, you say absence of centrum in atlas. Here you write presence of large and thick or massive centrum. Then there are other differences. You can say metapophysis here absent, that is in atlas. Metapophysis in lumbar present. And also there are other differences if you check well. This one also have a hypophysis. Don't have hypo. That is atlas don't have hypophysis and apophysis, but is present in lumbar vertebra. So that is the thing. Then this one have reduced neural spine. This one have a pronounced neural spine. That is your lumbar vertebra. The neural spine is not reduced, but in atlas the neural spine is reduced. Then here you say state two similarities between B and D. Specimen B is thoracic vertebra and D is lumbar vertebra. Presence of neural spine or presence of spinous process. Presence of neural canal in both. Presence of a centrum in both. So both, there are many things you can put there. Lumbar vertebra and thoracic vertebra all have, both possesses spinous process or neural spine, both possesses neural canal, both possesses centrum, both possesses transverse process. So that is it. We are done with this. I've given you many options, but let me fill this one just for formality's sake. Okay. So here I said you can write presence of vertebratorial canal. Okay, the thickness is much. Presence of vertebratorial canal. Over here you say vertebratorial canal absent. Vertebratorial canal, of course, is absent. Then secondly, centrum absent. Here centrum present, or body. Don't call it centrum, you can call it body. And remember that difference I told you. You can also say metapophysis absent in A but present in D. Then two similarities between lumbar and thoracic is that both possesses neural spine, presence of a neural canal in both, So there are other ones. We go to the next page. You have settled this one. Okay, I think this is page number 3, and the question is say draw a well labeled diagram 8 to 10 cm long of the posterior view of C, and the specimen C is actually scapula. So posteriorly, you should watch down my video there and see how to make the view. Posterior view, like you are viewing it, what they mean here is actually from the back. So when you view from the back, that's where you show the, remember in this drawing you are required to show the neural spine. You are required to show the spine of the scapula. That's one major thing. That's why they say posteriorly, because in the anterior view of it or the costal surface of it don't have the spine which projects up. Then the rules, you have to show the glenoid cavity. These are the things that will give you mark. Remember, in biological drawing, you are not just drawing anything you like. Maybe I'll bring a separate note for it. So here they said 8 to 10. Maybe I'll highlight my electronic ruler here. Okay. Over here we have the ruler. With this, you can mark your 8 cm, meaning if you draw more than 8, no problem. 9, okay. 10 cm, okay. But when you make it 12, you are off the range. And size of your drawing have a mark also, so don't joke with that size. And as you can see, I think this is no more than 8 in the real paper, in no more than 10, sorry. So you make your drawing as much as you can to contain in this space given. So since I'm answering this, no need of even going to that drawing sheet. Let me answer it here direct. In the scapula, okay. The problem here is that this pen is red, so although I can still make it black and reduce the tone. Okay, so I think they are making it this size. It's okay. So bring our sheet here. A scapula is triangular plate. Make it this way. Which extends in this way. That of rabbit is what I'm making a sketch of. So at the end you might have your spur of scapula. So remember my lines are wavy, so I have to okay. So in labeling, this is actually the, depending on, you just label the plate bone. Here is the coracoid process, and here is the glenoid cavity or glenoid fossa, and here is the spine. You need to indicate the spine, the supra of scapula. Then on top of this drawing, you must not forget that you have to give it caption, which is, according to them, they said posterior view of the scapula, or you just say specimen C. It has a mark, posterior view of scapula. So title, when you write in capital letters, you don't need to underline, but when you write in small letters, you underline it. Then here is our coracoid process, coracoid, to give you mark. Here is the acromion process, and label one side, acromion process. I think I have to use pen too, or is it clear? I think it's a bit clearer. Acromion process. Then here is the metacromion process, metacromion process. Then this glenoid cavity is very necessary. It's one of the things that will give you mark. You must show it the spine, since they say posterior. Then don't forget to write magnification, mg, below. So your magnification is this drawing. You can use ×1, yes, of course. You can use half or 1. 1 means you drew exactly the size that was provided for you. So it's possible that the rabbit scapula is up to 8 cm. So magnification is actually equal to the size of your drawing all over the size of the specimen provided. So, but you are not going to do any mathematics here. It is just a necessary formality, and use your discretion when you are making drawings of something that is smaller than the real object, the real specimen. The magnification is going to be actually less than 1. But when your drawing is bigger than the real object, your magnification is going to be more than 1. It's just asking you how many times you have magnified the drawing. So this is the structure of the scapula, and the labeling settled. Then the next one is identify specimen E, F, G, and H. You lift our this thing here again. Simple. It will be provided with them, but here I don't have them. E is a putter, so I'll go back and pick my pen again. Specimen E is putter. Remember, you are not allowed to write with red pen. I'm writing with red here for clarity. Then specimen F is sweep net. Specimen G is tridax fruit. So specimen H is copy direct, third pole. Okay. The now says state the function of E and F in ecological study. Specimen E is actually a putter. F is also a sweep net. I think two of them have the same use in ecological study. It is used for collection of insects. Collection of insects. This one is also for collection of insects. Collection of insects. So the difference here is that putter is actually used to collect very small sized insects like the ants. You use it to suck in the thing. It's like an aspirator. The putter works like an aspirator, but sweep net use it to collect many of them, like if you want to collect crayfish from water. So but two of them are actually used for collecting or collection of insects. Then state the mode of dispersal of G. And specimen G is tridax fruit. Is dispersed by wind. Simple. If it might interest you, it is dispersed by wind because of the fact that it has a membranous wing. The fruit wall is modified as to form a membranous wing. Okay, sorry, not for tridax, please. Tridax, the fruit wall is modified to form a parachute. Yes, a parachute, which helps in buoyancy and propelling in air. So now we have number 3, number IV here. Just says list 3 morphological adaptations of H to aquatic life. Specimen H is tadpole. Morphological adaptations, you take a look at the shape. That is externally, it is streamlined in shape, of course. You know, morphology is actually shape. So it is streamlined in shape. Then presence of tail for swimming. Then presence of external gill for gaseous exchange. These are the 3 things that adapt tadpole to survive in water. So you can say streamline shape to reduce drag. Then another one is presence of external gills for gaseous exchange. Then presence of tail for swimming. Presence of tail for effective swimming. So, but they just say list 3 morphological adaptations. So, streamline in shape to reduce drag, presence of external gills for gaseous exchange, presence of tail for swimming. So we go to next page. We are done with page 3. Remember, you are on with Her Majesty's Easy World Science channel. Okay, this number page 4. Actually, we continue from question number 2. Here it says identify specimen I, J, K, L, and M. Simple. You can see that I is Pyrogyra, J is okra fruit, K is mango fruit, L is teak, then M is beanweevil. So they say using hand lens to observe I, state 2 of its features. That should be Spirogyra. With the aid of hand lens, you should observe that it is filamentous. That's it. That's one thing it will reveal to you. So here you write it is filamentous. Of a cellular cell wall. That's why it's greenish. Then here it says in a tabular form states 3 differences between J and K. That is differences between okra and mango. J first is a dried fruit. This one is a fleshy fruit. or succulent fruit, fleshy fruit. Then many lines of split present. Lines of split present. Let me minimize this. Lines of split present. Lines of split absent. Or you say that this is dehiscent and this is indehiscent. It is still correct. Then this one you can say is a capsule, then this one is a drupe. So because the differences, they say they did not limit it to observable. It can be any difference among them that you know. Any of the differences among them that you know, you write down there. That is okra. Remember, okra is a dry dehiscent fruit and the mango is a drupe. It's an indehiscent fruit. Then you can even say that this one has stony endocap, absence of stony endocap. Then the placentation, this one have axile placentation. Axile placentation, and this one is having marginal placentation. Then the next one is name the class in each to which L and M belong. L is a tick, belongs to the class Arachnida. Then M is an insecta, pinweevil. That's the class. So the other one says states one economic importance of L and M. Of course, tick is an ectoparasite of most mammals. Of course, is an ectoparasite. Then the same thing with weevil is a storage pest. Storage pests attacking grains. Of course, storage pests for farmers, of course. Then that's it for this page. Over here is page 5. So which says draw the longitudinal section of 8 to 10 cm long of specimen K. Longitudinal section of K, and K is a mango fruit. K is a mango fruit. So remember, you need to have your caption. It's compulsory, it gives you mark. Don't say they know what they asked you. You have to write it on top. Simple. Longitudinal section of mango fruit or longitudinal section of specimen K is a title there, which has its own mark. Then let's highlight our pen and change to pencil. Okay. Let me increase the thickness. Okay. Yeah, I think it's okay. Then in drawing of the mango, don't bother yourself. The little secret you need to do here is just make an oval shape, complete oval shape. Okay, remember to leave a space where you have to write the title, please. Longitudinal section of mango fruit. Okay, longitudinal section of mango fruit. Because I used small letter, I have to underline. Then in the drawing, what's the secret? Make an oval shape. Okay. So, stay in the cleaner. Take your pencil, please. So you make an oval shape like this. Remember to obey the size of your drawing. They say 8 to 10. you still leave I have to leave a small space, or I might write magnification here. For mango, you still use ×1. Nobody knows the size of mango that was provided, ×1. But the problem is using ×5 for mango is wrong. ×1 times 0.5, that's times half, is allowed. So this is not looking like mango. What you do, find the half. I think this is a half. Go above the half, then make a curve. Yeah, I think I'll still go higher than this. So go above the half, of course. Then you make a curve, and when you make this curve, you clean off this, you have a shape in this form. So you are very close to what you want. So in this case, you place your fruit stock, then double lines will be used here. Use your double line to show cross section. So there will be a layer known as epicarp, since they say longitudinal section. Then there is a second layer known as the mesocarp, which will be the largest in size. Then after that, we have now the seed. So that's the thing. Your work now is to label. Remember to write your mg, which is, I told you can use ×half, or you can use ×1 for mango. No shading is allowed. This is enough. You know, this my line ran together, so I'll probably clean off. Okay. Let me perfect my drawing. Nothing wrong with it, but let me make it look perfect. Neat drawing. Have his extra mark. Yeah, so in this case, in your labeling, you might label with this is the first layer. Make your lines of label very straight, and this is epicarp. Then here is the middle layer, and this is the mesocarp, which is the edible part. Your lines of label should be done with ruler. That straight lines of label has its own mark, so let me use ruler in this case. Let me use object cleaner so that yeah. Okay, so I got my line and my ruler here. Okay, keep it that way. This is the epicarp. That's the mesocarp. Then here is the endocarp. Then over there is the seed. Yeah. Then fruit stock can also be labeled. Now it comes to labeling. Let me use pen. So here is my fruit stock. Then here is my epicarp. Then here is my mesocarp. Make your spellings correct, please. Mesocarp. And here is the stony endocarp. And here is the seed. So we are done with this. Remember not to shade and use light lines and make it visible. Your caption is there. So why marking this? Biology is not just marked by ticking it good. You see them write something like SD, size of your drawing, MG, caption, that's title has its own, then proportional drawing showing the mesocarp being largest, and so on and so forth. So it depends on what the marker wants and how he wants the drawing to be developed, but there are basic things that must be shown. Okay. Number next says state two structural differences between L and M, and over here our L is thick and the M is being weevil. Structural differences. Here you need a hand lens to enable you make the difference, but first know that in L, which is a tick, you say antenna absent. Ticks don't have antenna because just like other arachnids, they lack antenna, and I think they are the group of arthropods that lacks antenna, the arachnids, that's the spider group, they don't have antenna. Then bean weevil is it's not an arachnid, so it is an insect, therefore it has a pair of antenna there, bean weevil. Then another difference is in tick, there is an absence of wings in ticks, then wings present in weevil. So you can use these two as a structural difference between them. That's structural differentiate them. Then structurally, tick has two body divisions, the head and the cephalothorax, while bean weevil have three body divisions, the head, the thorax, and abdomen. That makes it an insect. Structurally, the ticks also have 4 pairs of working legs because it's an arachnid, whereas the bean weevil, with the help of your hand lens, have 3 pairs of working legs, which makes it an insect. All insects have 3 pairs of working legs, making it 6. So all these things I've said, you can use any of them and differentiate. Remember, structurally is what they say. That's what you are seeing. Here, wings present in the bean weevil, absent in that of tick. You can also tell them body division into 2 in that of a tick, then in weevil divided into 3, antenna present in bean weevil, absent in that of a tick. I wouldn't know. Yeah, they should have a kind of a pair of antenna. Then identify specimen N, O, and P. Specimen N is cactus plant. You just fill it there. Cactus plant. Then specimen O is water lettuce. Then specimen P is soldier termite. Remember, they did not say identify with reason. Just write what you are provided with. So soldier termite. Some might not understand what I said here. Structural difference between tick and bean weevil. So I mentioned 1 body division. L being tick, 2 body divisions. 2 body divisions. This one 3 body divisions. Then there is a structurally in the bean weevil, there is also this one structurally have 4 pairs of working legs, 4 pairs of legs, and this one have 3 pairs of legs. Okay, the mandible present. Okay, no, none of them have mandible actually. Then okay, those 2 are enough. Then we'll go to the next page. We are done with this page 4. I hope so. Page 5. Okay, we're almost through. Save my time. See the answer here. Okay, page 6 says mention 1 habitat each of N, O, and P. Habitat of N is N itself is a cactus. You write desert. You can find it in desert. So it's a desert plant. In arid environments, it survives there. That's where they are commonly found. Then O is water lettuce. It's found in you can use found in aquatic habitats, of course. In aquatic habitats, usually freshwater habitat like river. Then P is soldier termite. It's found in a termite mold or anthill, termitorum, depending on termitorum. You can find it in wood, of course. That's the places they can occur. They can also be found in wood. There are many things that can serve as an answer here. Then next one says mention 3 adaptive features each of N and O that enables them survive in their habitats. N still remain cactus plant. Features that enables, you say it has leaves reduced to spines. Leaves reduced to spines or hairy structures. Then thick succulent stem for proper storage of water. Thick succulent stem. Then again, it has a sucking stomata. Yes, it has a presence of numerous stomata or sucking stomata, kind of hidden stomata there. Stomata. So then O is specimen O is water lettuce. What enables structures that enables the survival of water lettuce is presence of hairs. Presence of hairs in the leaves and also the roots. The leaves and the roots to enable buoyancy. That one is flammable. Then waxy, the leaves are waxy leaves, to actually repel, for water repellent, so that water can easily fall off. Then number 3 is let's be logical here. That's broadleaves, of course. That broadleaves with numerous stomata. Broadleaves with numerous stomata to ensure effective transpiration. You know it suffers excess water because it's in water, so it adapts a means of effective transpiration. Then draw, the last one says draw a well-labeled drawing of 8 to 10 cm dorsal view of specimen P, and specimen P here is soldier termite. So in the soldier termite, they say well labeled 8 to 10. You still obey your dimensions in drawing. I got my pencil here. Okay, so in this case I might give you a clue that will help you in the drawing. First, you have measured, remember your title first, you must give a title. Just write dorsal view of soldier termite or specimen P. Dorsal view, let me use sticker. Oh, sorry. So dorsal view of soldier termite. So that's it. Then, because they are in small letters, I'll underline. I want to reduce this intensity because you have to draw with not thick lines, light lines, of course. So in this case, I'll give you an easy cheat on the drawing. First, you might make something like W like this. Then on top of the W, you write U, like you're writing capital U. Okay, I have to bring it down again to make it contain there. Okay. So here you make a kind of what's wrong? Partial W. Then on top of it, why making a line for me? I don't like that. Yeah, that's your U. Then on top of this, you create more, and you surround it with sharp mandibles for biting and chewing. And here you surround it with your maxillary pap. Then after which you now draw your antenna, which is bigger. Segment it because the antenna is joint. Then here there will be kind of 3 layers in the thoracic. Then you make it this way. At the end you have your soldier termite. Put your circles here, put your circles here. Okay. Then in each of these segments you have your femur fixed. Then you have your tibia. When you fix the tibia, you fix your tarsus. And each segment like this, the same way. Have the tibia still, fix your tarsus. So you might decide to bring this one down. Femur, tibia, the tarsus. So in the same way, then don't forget to put your labial, because they say detail labeling should be there. Tibia, tarsus, jointed more. Okay. So then, don't forget to segment your drawing. So this is it. You have your job done. Then mg for termites supposed to use times 10 because it's too tiny, such that 8 to 10. No soldier termite, don't use times 1. This drawing shows a magnification that is not less than times 5. Of course, you can use times 10 or times 8 depending, but don't use times 1 or times 2. That becomes wrong. Then in the labeling, remember to use your ruler. I might highlight the already labeled ones here. Yeah, see this? But this is not actually depicting the very big head of soldier termite. Make sure that the head will be bigger than the rest of the body. That makes it a soldier termite. This looks like a worker, actually ordinary worker case in that. So you can see another one. I think this is a bit very correct. The head is too big. Make sure your own head is very big. This is the one I drew with this app in a relaxed mood. So in your labeling, you make your lines as usual. Use my ruler here. Here is the antenna. Here is the mandible, sharp mandible. Here is the maxillary pulp. Here is the labial. Okay, that's too close. I'll clean that one. Then over here is my, so this is allowed, the labium, labrum, of course labrum. Then here is the head. Here is the thorax. The lines of label should be straight. Then here is the jointed legs. You might label separate. Here is the femur. Here is now the tibia, and here is tarsus. Then here is your abdomen, and here is our circus. See that? So I'm done with the ruler. I'll bring back my this. Okay, clean where necessary. You don't need this. Let me use this. Then make it as equal as possible. Then you start your labeling. Start putting those things that are there. Let me use a blue. Intensity is high. Use here. So here is antenna. See it there. Remember, spelling matters a lot. Antenna. Here is the mandible, sharp mandible. Here is the maxillary pulp, maxillary pulp. Then here is the labrum. Here is the head, and here is meant to be the thorax. I'll point the arrow well. And here is meant to be the femur, the coxa. Then here is the tibia, and here is the tarsus, and here is abdomen, and here is the circus. So we are done with this particular question for the year. So whenever you finish up with this, you get your job done. So God bless you, and remember that this is Her Majesty's Easy World Science channel. We're here for you. Apart from exams, there are other things we hold on to here in this channel. It's not only about exams, as most of you remember us here only during exams. There are other entertainments we offer, as you can see here in this video.
I'm here to encourage you to subscribe to Her Majesty Easy World. He does fun experiments and science missions. Please subscribe, and yeah, have a good time.
Extrinsic muscles of the eyeball. Different actions of the latissimus dorsi.
There you will find a lot of amazing videos, and yeah, they are very educative. And subscription is free. Just go online and hit on the button. Thanks.
About 50 videos are available for you. Educative and informative, inspiring and entertaining industrial production processes. Her Majesty Easy World is an online school of science. No school fees, no stress. We teach the real thing. Thank you for being there. Remember to subscribe and share these videos and ask your questions in the comment box. God bless you. I love to know the fact that you are there for me. Bye for now. I love you. Thank you for watching.
Alpha and Omega.